Hi, my name is Altaf. I'm a postbac researcher at Reed College and an incoming PhD student at CU Boulder.
I am Oliver Anderson. I'm also a postbac researcher at Reed College. And today we're going to be presenting GRPhIN, which is our new tool. So to start out, essentially just do a basic overview of graphs. Graphs are ways to model complex systems, especially within biology. So two examples of graphs that you may be familiar with are gene regulatory networks and protein-protein interaction networks. So these have very similar nodes. In gene regulatory networks, nodes represent transcription factors or genes, and there are directed edges indicating regulation. In protein-protein interaction networks, nodes are proteins, and the edges are undirected physical association or binding events. So they have very similar node representations, but different edge representations. So this may potentially obscure the full functional context of a protein because proteins, genes, and transcription factors are not separate entities. Proteins act as transcription factors, genes encode proteins, and also physical binding events have been shown to have regulatory effects. So by keeping these graphs in isolation, you may actually obscure the full functional context of a protein of interest. So to combat this, mixed graphs come in, and these allow you to have multiple edge types with the same node type in a graph. So what can we do with mixed graphs? One thing that we can do with mixed graphs is look at essentially local patterns within the network to characterize the network in some way. So one of these local patterns that we're looking at are called graphlets. These are essentially small connected patterns of n nodes that are basically an induced subnetwork that describe the local organization within a graph. So in this figure over here, these are all of the graphs in an undirected, or all the graphlets in an undirected graph from 2 to 5 nodes. And furthermore, within a graphlet, you can also characterize the role a node plays, and these are called orbits. So in G1 over here, we have 2 actually distinct orbits because the edge nodes are occupying the same role relative to the rest of the graphlet. So orbits and graphlets have a variety of different applications. Specifically, counting graphlets and orbits can provide a lot of information about a system. They've been used for protein function prediction, characterizing like the differences between disease and healthy states, among various other things. So this is where GRPhIN kind of comes in. We were inspired initially by a paper from 2004 by Jaeger-Lodem et al., which basically combined a regulatory network and a PPI network into a mixed regulatory and physical network, and counted all of the graphlets in yeast. So we wanted to sort of take this idea and make a generalizable tool that could be applied to your own PPI or regulatory inputs. So we looked around for different algorithms to see if we could do this, and we found one called Orca, which is very efficient, but it only works on one edge type. Furthermore, we also were inspired by this paper by Somnes and Khan, which did something very similar to what we did, but they counted a very specific network, counted graphlets in a very specific network with many different undirected and directed edge types, and we also could not find like open source code to apply this to our networks. So we kind of wanted to combine all of these gaps together and create a tool that could work with not just GRNs or PPIs, but any sort of undirected and directed edge inputs that have the same node representation. So we're presenting GRPhIN, which enumerates all 2 and 3 node graphlets in a mixed graph. And this is a little overview of what's to come. We're gonna apply GRPhIN to 5 different species. We're going to do a graphlet over representation analysis in stress response subnetworks for each species, and an orbit over representation analysis in oxidative stress response proteins. And so this is sort of to identify mixed graphlets and orbits within and across these species that might be indicative of oxidative stress response, because oxidative stress response is like a pretty conserved pathway across all species. Like to survive on Earth, you have to deal with oxidative stress.
Yeah, so before going through the GRPhIN algorithm tool, we just wanted to iterate sort of the counting problem that we have in graphlets. For example, depending on the configuration of your network, you can have different numbers of graphlets. For For example, in the 2-node undirected graph, there is only one graphlet count, as you can see there. For the 2-node directed graph, there are 2 different graphlets. And for the one that we're interested in, we have the mixed graph for the 2 nodes, and there's 5 different configurations. And again, this is not so bad in terms of counting this across a large species networks. But what about the 3-node graphs? For the 3-node undirected graph, there is 2. You have a line and a triangle. And for the 3-node directed, there's 13. But what about the mixed graph? Unfortunately, there's 98 different configurations. And so the jump between 2-node to 3-node for the mixed graphlet configuration is pretty massive. And this is going to be computationally intensive when we apply GRPhIN on our later biological networks. And so before talking about, after talking about the problems when it comes to counting graphlets, we wanted to be able to efficiently create a tool that would be able to count all the graphlets in a biological species networks. And before talking about that, we wanted to start about characterizing edges. And this is really inspired by the paper by Somais and Can, which uses a binary vector to represent the multi-labeled edges. So in our case, we used a 2-node graphlet characterize a 3-bit vector for the undirected edge and also the directed edge. And as you can see here on the figure, there are 8 different encodings that we can use for the 8 different edge types in a 2-node graphlet. So after talking about the 2-node graphlets, we can apply this to characterize 3-node graphlets. And we'll be keeping track of this example and also the edge encodings on the right side. And we can actually break things down into their specific nodes. And we can learn about their structures and how they connect with each other. So for example, in the node A, it has at most 2 encodings that goes from A to B and A to C, and those are 1 and 4. And if we do the same for B, B to A, B to C, it is 1 and 4. And if you notice that they both make the same integer pairs. And if we look at C, C to A, C to B, there's 5 and 5. And so we actually get this really interesting structure that we can represent how we define 3-node graphlets in a mixed networks based on all the nodes and its relationships within each other's. And so for the sake of our presentation, all of our tools available on our repo that we'll plug in the end. And we do a few more things related to breaking symmetries and sorting, things like that. And so previously mentioned, the counting part is kind of a difficult part. And we use like an enumeration approach, which iterates through all the nodes to find the triple of nodes. And this is computationally intensive, as we found. Actually counting the graphlets was not that bad, but it's the amount of times we have to do lookups in terms of like the edge properties. It added a lot of like runtime, and yes. And checking for isomorphisms and breaking symmetries also most definitely slowed things down. And now that we've introduced GRPhIN, we kind of wanted to apply it to 5 different species networks. And this is how we kind of collected our data. We use 5 different non-human species. As you can see here on the table, we have varying sizes in terms of interaction types. The largest network that we have is the yeast, which has almost 500,000 or 400,000 edges in total. And they're collected from species-specific databases and general databases. And all of these files are available in our repository if you'd like to use it in other projects or in GRPhIN. And the other part of our project that we're interested in is looking at the relation to responsive oxidative stress. And we can find all of these proteins that are associated in all of these organisms through the geontology term called a response to oxidative stress. And we can see that there's varying number of stress proteins associated for each organism. And so we wanted to take this to see kind of like what are the distributions of graphlet among non-human species. And to illustrate just one example, we used the D-melanogaster to illustrate all the 3-node graphlet counts. And yeah, the red line is all of the top 10 graphlets that are across in all the species. And furthermore, we wanted to focus in on the specific mixed graphlets that we talked about before. And we found that these 3 graphlets are in the top 10 graphlets across all of the species. And we thought that was kind of interesting. Yeah.
Yeah, so kind of taking those stress proteins, we wanted to generate a single connected network that we thought would represent oxidative stress response. So to do that, we took these stress proteins and ran a random walk with restart on the underlying network with the many thousands of edges, and restarting at these stress proteins. So this generated a network with the stress proteins and also some surrounding proteins that was connected. So from there, we ran GRPhIN on that, and we also generated 1,000 randomized networks holding the same structure as the stress response network. And so to do that, we essentially, looking at the multi-edges, we preserve the ingoing and outgoing degree for every node by basically generating a switch condition that allowed you to switch edges between 4 nodes. So doing so, we kept the structure of the networks, but we sort of scrambled up the biological significance. From there, we calculated whether or not the observed counts of graphlets in our real stress response subnetwork was surprising compared to the 1,000 randomized networks. So these are some results. We wanted to focus in on the mixed graphlets because that's sort of the novelty of our tool. And what's interesting here is that we actually see this. I'll describe this graphic or this image a bit. This is the, I guess, most common, so most frequent count, significant overrepresented mixed graphlets within each species. And so we wanted to sort of focus in on one of these as like a case study to sort of maybe speculate as to the biological significance of this. And so we focused on this one because it was present in 4 of the species. And we sort of took a hypothesis that this is a transcription factor that regulates 2 genes whose protein products physically interact. And so we took a look at this graphlet within the B subtlest network because it was the smallest and easiest to visualize. And these yellow nodes are all part of this graphlet, but we wanted to sort of focus in on just one for the purpose of this presentation. This graphlet right here shows sigma factor A, which is a generalized transcription factor. And it's actually regulating 2 genes, AHPC and HPF, which certainly do physically bind with each other. And they're involved in detoxifying free radicals during oxidative stress. So it's sort of like a sanity check that our networks that we generated were good. But this sort of highlights the ability for GRPhIN to pick up patterns that traditional or previous methods that work on only a single edge type would not be able to pick up.
So after looking at the graphlet overrepresentation in the stress networks, we want to look specifically in the orbit overrepresentation. So to clarify, orbits can be defined as the different roles that a node participates in the context of a graphlet. This is the previous example we looked at. Visually, you can see that node A and node B kind of have the same role in terms of they have the same outgoing and incoming edges. And node C has a different role, and they're indicated by the different colors. But if we look at the previous definition of how we characterize a 3-node graphlet, we can see that node A and node B actually have the same integer pairs. And so we can see that all orbits in a 3-node graphlet correspond to a unique pair of integers in the context of its graphlet. And we compiled a list of all the stress-related proteins previously, and we were inspired by this paper by Agarwal et al. At 2018. And it really asked the question of if stress-related proteins occupied a certain orbit. And so our method that we did was compare non-stress proteins versus stress proteins on orbits to see if one is overrepresented over the other, given a certain p-value. And for more details, the method's available in our repository. And this is kind of a breakdown of all the different stress proteins for each organism. And we found that 4 out of the 5 organisms had overrepresented orbits, and the most being in the fly organism. And we found that also 2 of them were conserved across all species. So these are specifically orbit 1 and orbit 7. And while they're not, unfortunately, mixed graphlets, they're just kind of regular graphlets. Yeah. And so GRPhIN's pretty much available through our repository. And this is kind of a general workflow system. While we focus on PPI and GRNs as the inputs for the undirected network and the directed network, we hope that this can be applied to over other areas of science. And it is able to enumerate all 2 to 3 node graphlets and generate an output file that hopes that you can do more post-process analysis, similar to what Orca does for further analysis. And some of the general next steps we do is most definitely the enumeration process and scaling to more nodes types, and yeah, implementing it in another system, and specifically diving into the specific biological context of mixed graphlets. And yeah, thank you so much for talking in our presentation. This is our link to our GitHub repository. And yeah,
Thank you.
thank you.
Do we have any questions? Feel free to come use the mic.
Okay. Yeah, thanks. Really nice talk. Very cool app. So it made me wonder, were any of the results you have suggest that you might be able to prune the 3-node space in terms of those that appear to be biologically relevant, and maybe use those as cores for expanding to this kind of pruned or restricted set of 4-node graphlets?
Yeah, I think that's definitely one of the, there was a paper that showed kind of the significance of a lot of these other papers. Specifically, I think the Jaeger-Lodom et al. Focused specifically on the 3-node, and they've been kind of shown to be computationally like a middle ground to focus on. But I think definitely for 4 nodes, that's kind of the next step. And unfortunately, we don't have sort of the next steps to kind of do with that. But I think further research, I think, would be great for that.